So yeah, welcome back. I think my voice is clear, and after a long time, we're gonna today start the chapter cells. So yeah, let's go. Let's start. So this time I'll not forget to share my screen. So yeah, first of all, just from the basics, yeah. So what is the cell? Now you're gonna be like, yo, we have studied this in O levels, and you can say 9th class, 8th grade, you can say that. So we have learned in our previous classes that cell is the basic unit of life. So this is the definition that we all have been memorizing since our childhood, yeah? So we're gonna modify a bit here. So it would be better if you start describing cell as the basic structural and functional unit of life. Sorry. So this can be a modified definition. Definitions obviously does not come in the A levels, but you need to, just for basic knowledge, this can be the new definition. Obviously definitions are perspectival, yeah, everyone have his own definition. It's basically how you explain a certain concept. So I would like to explain this concept of cells this way. Anyways, let's just move on. So cells were first discovered by Robert Hooke when he was playing with his microscope, just kidding, and he saw cork structure, yeah, which he then defined as cell and blah blah blah. These are some basic things that you obviously know here. Anyways, let's just start. There are two different types of cells that we're going to be studying. So two different types. And first we need to examine the differences between them. So first are prokaryotic cells and another one are eukaryotic cells. Now, what are prokaryotic cells and what are eukaryotic cells? So prokaryotic cells are basically found in bacteria, and obviously this is not what prokaryotic cells are, but I'm telling you for the reference here that prokaryotic cells are the type of cells which are found in the bacteria, archaebacteria, never mind. They have a bit different structure. We're going to be studying that concept in the chapter of, you can say, ecology, classification, sorry. We're going to be studying about that and classifications, but for the reference it's kind of like prokaryotic cells. And the eukaryotic cells are the basic cells that humans and plants, etc. have. So the key difference between prokaryotic cell and eukaryotic cell is a structural difference, yeah. So prokaryotic cell is much simpler than the eukaryotic cell, and eukaryotic cell has more components, as intercellular membranes, has intercellular compartments, which you can say organelles. So the basic and key difference between prokaryotic cell and eukaryotic cell is that prokaryotic cells does not contains any organelles. And what do I mean by organelles? We're gonna be exploring that in a bit. And eukaryotic cell does contain organelles. So what are organelles here? So you might have heard about mitochondria, or you can say you can say about chloroplast, or you can say nucleus. So these things that are enclosed in a membrane. Remember what we learned about phospholipid, that they form a membrane, they form a structure when they are in water, yeah? So this structure that they form, the membrane encloses, is called organelles. So anything found in a cell which is enclosed in a bilipid membrane is gonna be most probably called an organelle. So all of these, mitochondria, chloroplast, nucleus, are either way, might be double membrane or single membrane, are enclosed by a membrane, giving them separate compartments. Yeah. So the basic difference between prokaryotic cell and eukaryotic cell is going to be that prokaryotic cell is one compartment in itself, and eukaryotic cells, due to its complexity, contains different compartments in itself. So I'm going to be listing down different, you can say, differences here. So for example, nuclear membrane is not present here, so the DNA is naked into the cytoplasm, you can say, the plasm that is present in the cell. So nuclear membrane doesn't present. That means literally that even the DNA of it does not have a separate compartment here. It contains that. So vice versa is present. DNA is naked and exposed, which is definitely this due to the absence of nuclear membrane. So we're not going to be listing down that as a point, but you can memorize it. Because nuclear membrane is not present, DNA is naked, exposed, scattered around in the cytoplasm. And within the eukaryotes, DNA is enclosed or surrounded by membrane. Also, it does not contain so the prokaryotic cells does not contain any membrane bound organelles, so it does not have membrane in itself. So whereas the eukaryotic cells contains membrane bound organelles, different compartments within the cell membrane of the cell. So another thing that we're going to be, another difference is going to be that DNA in prokaryotic cells is circular, which is in cytoplasm obviously, whereas in the eukaryotic cells DNA is linear. And what do I mean by that? We're gonna be looking in a bit. Now, the statement that I wrote in the first I have written that DNA is found in linear form in the cell, in the eukaryotic cells. But the second line is but circular is also present, and it will make more sense ahead when I show you the diagram. Yeah, so just leave it for a minute. Another big difference here is in prokaryotic cells, cell wall is made of a compound of complex protein which is called peptidoglycan. Or you can say murine. This is the second name of it, nickname you can say, if the peptidoglycan word is hard for you to memorize. And in the eukaryotic cells, there are different cells in which cell wall is not present, and some cells in which cell wall is present. And the cells in which the cell wall is present are usually made up of cellulose, is either present or not, and if present, sorry for my handwriting here, it's made of cellulose, which is the polymer of the beta glucose, as we discussed in the biological molecules. So chromatin is absent in the prokaryotic cells. And what do I mean by chromatin here? So chromatin is, as I have told you before in the biological molecules about the conjugate molecules, nucleoprotein. So chromatin is simply a second word for nucleoprotein, yeah, because chromatin is formed from the combination of DNA plus a histone protein, yeah. So for example, this is DNA. This is DNA in form of chromatin, yeah. When it combines, it usually wraps up around like a bead-like structure, which is usually a protein, yeah. So the structure on which it coils, the protein, it's called histone, and the combination of them forms the chromatin, or you can say the chromatin material. So it is formed from DNA and a histone protein. Another major difference, so we should write here chromatin present. Anyways, another major difference here is only 70S ribosomes present. Whereas here both 70S and 80S are present. Now, here we got mesosomes in the prokaryotic cells, whereas mesosomes are absent in the eukaryotic cells. Now, most of the prokaryotic cells would contain locomotory structures because they are on their own most of the time. they are single cellular structures, they need to move, yeah. So those locomotory structure, for example, pili and flagella, are present in most of prokaryotes. And few eukaryotes because they are mostly found in colonies or piled up, so we'll just see about that ahead. But anyways, so these are major differences between a prokaryotic cell and a eukaryotic cell. Now, we need to talk about eukaryotic cell. We need to dissect it. We need to take out all the different types of organelles. During those times, I'm going to be explaining how does the processes that are occurring in the eukaryotic cell occurs in the prokaryotic cell, yeah? So, let me just divide the structures found in a eukaryotic cell. So we can divide on, or we can classify on the basis of double membrane structures, or you can say single membrane structures. Double membrane structure and single membrane structures. And some structures are no membrane. So in double membrane structures, some of the most important organelles that are the basics or basis of our cell's survival are nucleus, this which is double membrane structure, mitochondria or mitochondrion, whatever you can use. Another one is chloroplast, and you can say cytoplasm, which is technically inside the cell, so it is enclosed. No, sorry, cytoplasm definitely not double membrane. Anyways, single membrane structure, endoplasmic reticulum, ER, which would include RER. I mean, ER is based on two different structures, RER and SER. So we're going to be differentiating between them too. It also contains lysosomes. It also contains Golgi body or Golgi apparatus, glycosomes, glyoxysomes, and different types are also single membrane. And etc. I'm going to be giving a brief overview on the different single membrane structures which are not present in A levels but are present in MCAT structures. So I'm going to be just giving a brief overview of them and at the end. Anyways, the no membrane structures are basically ribosomes and nucleolus. And centrioles, which are only in animals. So these all you need to remember, because a lot of times the questions in A levels comes from this particular information. They're going to ask how many membranes are there, single, double, triple, and they're going to ask about nuclear loss, ribosomes, centrioles, and etc. So you need to memorize this very thoroughly. Anyways, let's draw a diagram of the cells. And 1, 2, 3, boom, here we go. I've drawn this in the past, but so I mean it would have taken a lot of time, so I just import it here. And here we are. This is a typical eukaryotic cell. Now we need to label it. We first label it, then I'm gonna explain it bit by bit, and I'm gonna for your ease, I am gonna you can say mention which are exceptions in plants, which are exception in animals, so it's gonna be easy for you to memorize. Anyways, you need to know every single detail in this cell, and you need to know its function, its name, its structure. It's very important. One thing I forgot to draw here is this, which I'll explain what it is in a bit. Anyways, this is a typical cell. First, we have this giant structure, which is cell wall, and this is typically in fungi, Protoctista, plants, and anyways, these two are, you need to know these two for the AS levels, but in A levels they are going to be Protoctista and another types or classificate classes, but typically cell wall you need to associate with fungi, plants. The second structure that comes is lipid bilayer membrane, or you can say cell membrane, cell surface membrane. There are different names for it, but this is the most common. And this is found in both animals and plants. So this is going to be mnemonic for animals in class A and P. Anyways, so these two structures. The third one is these dots that are spread all over the cell. So these cells, these structures are called ribosomes, and these are present in both. Or maybe I should change the color of the ones that are found in plants. So for your ease, the ones that are found only in plants are going to be in green. Yeah. So orange ones are only found in plants. Yeah. Whereas the ones in red are only found in animals. So ribosomes are, and the ones that are found in both are going to be with the black. So these are ribosomes, and the, hold on. Yeah, sorry. Oh. So the empty structure, the empty part is not empty at all. It contains cellular matrix, you can say the liquid part, which is cytoplasm, which contains minerals, which is basically the living part, you can say. It contains everything it is a medium for chemical reactions, and it is mostly composed of water. Yeah, it contains, I think, 95%. No, I think 70 to 80% water in itself. Hold on. Let me just confirm this. It's like, I think 95% water, but I'll just confirm it in a bit. Anyways, this is cytoplasm, a medium for the reactions to occur, and it contains every single organelle in itself. Plasm. So there's a difference between cytoplasm and cytosol. So cytosol is the starting part, yeah. So near to cell surface membrane, it's called cytosol, and as you go inside and towards the nucleus, it's gonna be called cytoplasm. There's a bit difference in them, but I don't think so you need to know about that. Anyways, this structure you see here, this is called mitochondria, the powerhouse of the cell. Another one, this structure right here, this is called ER, endoplasmic reticulum. These green ones, for your ease, I already colored them. So these are called chloroplast. The ones here we are looking at so these structures that we are having here are called lysosomes. And the structures that we have here, these cylinders, are called centrioles. Now there are different words. Centrosomes too is also used, but there is a technical difference which I will understand, which I'll explain it in the part where I explain centrioles. Anyways, this is called Golgi body or Golgi apparatus. Behind the name of a famous scientist from University di Pavia, where I want to go. Anyways, the black part here is this complete structure here is, hold on, I should just label it here. So this is called nucleus. And the part inside it is called nucleolus. And these thread-like structures, okay, we should not just label the organelles the intra-organelle structures right now, but anyways, so I think everything is okay. One thing I forgot here. These are only present, centrioles are only present in animals, so they are labeled with red. Okay, so yeah. So I think everything is covered here. The circles that you see here that are not labeled, they are not basically double membrane structures, but for the differentiation between lysosomes, I have created or given them double circles, but they are not double membrane structure. These ones I'm talking about, these, and they are glyoxysomes, peroxisomes, which I'm going to be explaining, but due to its absence in the syllabus of A levels, I have not made it a subject. I've not made it a topic. I'm going to be just overviewing it in brief. Anyways, so we are gonna be starting first with the prokaryotes. Oh, sorry, prokaryotes. I'm sorry. So we're gonna be starting first with the mitochondria. Let's just take this this structure here, paste it here, and zoom it. So this is a typical mitochondrion structure. You need to memorize the structure, you need to memorize the components that are present in it. So let's just label it. This is the mitochondria. Remember, I told you that it is a double membrane structure. So this is first membrane, or you can say primary membrane, or you can say outer membrane. So there is a difference between the permeability of both. Permeability means basically the allowance, the main function of membrane is to allow specific and reject specific materials to enter into the cell. So the permeability, which is the allowance of outer membrane, is much more than the inner membrane, as the inner membrane is much more specific in the passing of the substances. So it's very less permeable relative to the outer membrane. So intermembrane, this is intermembrane. The structure is called matrix. Anyways, these dots are 70S ribosomes. I should mention here that these one are 80S and these one are 70S, and the circle structures are circular DNA. And the space between the outer membrane and inner membrane is called intermembrane structure. Intermembrane space, you can say. Now, one thing you should have noticed here, if you have not, you're not concentrating much, my friend. So the thing that you should notice here, that the structure looks like or describes just like a typical prokaryotic cell, as we have learned about. So it has a lot of things common as of the prokaryotic cells. For example, it contains 70S ribosomes. Prokaryotes also contain 70S ribosomes. The second thing, It contains cellular DNA, circular DNA. It contains circular DNA, whereas the prokaryotic cells also contain circular DNA. It does not contain any membrane-bound organelles, so does the prokaryotic cells. Although it has double membrane structure, it does not have a cell wall. This is the only difference between them and the prokaryotic cells. But usually you can see that a lot of the properties of prokaryotic cells are similar to the you can say organelles inside these eukaryotic cells. So there was a theory that was proposed, the endosymbiotic theory, which you can say suggests that the structures that resembles the prokaryotic cells, for example, the mitochondria and you can say chloroplast, these two structures resembles the you can say prokaryotic cells, the properties of prokaryotic cells, as much that they are considered to be once a bacteria. They are considered to be once a bacteria. Yes, you heard me right. So the theory suggests that once upon a time, let me just give you a brief overview of that. So once upon a time, this is our eukaryotic cell. It was happy. No, I should just remove it. So once upon a time, this was a prokaryotic cell, which was happy. It was making food by itself, for itself. Now what happens is he meets a bigger fish here, a more dangerous, bigger eukaryotic cell. Or maybe I should, for the better representation, I should just fully represent it. So this was a happy living prokaryotic cell, which is, for example, for this scenario, you can say mitochondria. Now, one thing I should mention here is a lot of you guys might think that if we take out mitochondria out of a eukaryotic cell, it might survive. It's not going to happen nowadays because it has evolved into a more specifically a cell which cannot survive without the components present in eukaryotic cell. This is the power of evolution, but the theory suggests that this once was living happy, and this evil guy here, which is eukaryotic cell, which doesn't know how to make food for itself, comes and engulfs it, eats it. It gets into the cell of eukaryote because he was hungry. So now what happens is this eukaryotic cell is now a part of, and now inside the eukaryotic cell. So prokaryotic cell is inside eukaryotic cell. Now how will he get energy? There are two ways here we have. First way is the enzymes in eukaryotic cell comes to prokaryotic cells and kills it, and now kills the prokaryotic cell. Now after the death of prokaryotic cells, all the energy, ATPs, resources that the mitochondria, or in this case prokaryote, had, is now his, and he will now digest it and take and use all that for his energy. But now there is another way, a more friendly way, you can say. What happens is, instead of sending different enzymes and attacking the and killing, annihilating this, you can say demolishing the cell, what he does, he becomes happy too, and they make an agreement. They both agree to some certain things in which eukaryotic cells say Okay, we will engulf, we'll provide resources to you. You have to make food for us, or you have to make food for me. So here he says, I'll give you resources and protection, and you have to provide me food. Seems like a typical political system of the country, yeah? Anyways, but what happens here, mitochondria says, okay, you don't eat me, you don't digest me, I will have some autonomies for myself, I want to recreate, I will do it without your, you can say, permission, I will do it by myself, I can multiply by myself, I can do some processes that are within myself, I have to do it myself, you don't have to interfere, but I will provide you food, which is ATP in this case. So this agreement continues, and after that, these internal organelles, for example, the mitochondria and chloroplast starts living into the eukaryotic cell happily. And they have mutual benefit with each other, and because of this mutual benefit, this theory is called endosymbiotic theory. Anyways, we got very much into this. Anyways, we should continue with what is the function of mitochondria here. So for the record, we need to first, I should be just first telling you that the mitochondria is double membrane structure or double membrane organelle. Its outer membrane is more permeable than the inner one, and inner one is highly specific, highly impermeable membrane. So outer is more permeable than inner, which is highly impermeable. You should write it in your notes. Anyways, it synthesizes the basic thing, which is food, which we call ATP, the energy providing source within the cell. So it synthesizes ATP. And it has two compartments. You can see here, for example, this is one compartment here, as I can point at. This is one compartment, and this is another compartment here. So these two compartments, one is present in the matrix, and one is between intermembrane space. So it contains two of the compartments here. Now, the inner membrane contains different kind of foldings, which are called crista. This is the name of the foldings within the inner membrane. So this is extensively folded, and the reason for this is basically to improve the you can say surface area for more absorption, and we will just talk about the functions, why is this so much folded, what is the purpose of this in the chapter of bioenergetics, where we'll learn more about the processes that mitochondria goes through. Anyways, for this part, we're going to be I'm just going to be telling you that it is used for the transport, and when is transport going to be more efficient and fast? When there is more surface area, and because of more surface area, due to more surface area, the transportation is going to be more efficient. And for achieving this certain process, or you can say step, for high and efficient exchange between the both of compartments, there are extensive folding which increases the surface area. Anyways, increasing this due to this increase in surface area, as you know, in the past you have learned about this concept that surface area, the higher the surface area, the higher the rate of reaction. They are both directly proportional to each other. In a membrane extensively folded to accommodate more ATPase enzyme, so it is more folded because, for example, this, let me just give you an example. This is gonna be, so this is a membrane and it contains ATPase enzyme here, yeah. So this is, for example, ATPase enzyme. So this membrane is within the range of, for example, for this certain scenario, 10 micrometers, yeah. Now we take an example of the same, hold on, these same 10 micrometers. I'm going to copy it and paste it here, and the membrane now looks like this. And what happens is that the ATPase enzyme, because of the increased surface area, will get more space to be fit in the membrane. So because of the higher surface area, because of infoldings, the amount of ATPase enzyme will increase. What is ATPase enzyme? What does it do? It is none of our business right now. Just think it as the you can say structure which produces ATP. Anyways, mitochondria reproduces or divides by linear fission. This is the same, you can say, way how prokaryotes divide themselves. It's kind of similar, linear fission, and they are by independent of the nucleus. So the independent of cell means that it does not need dictation from the nucleus or any compartment or any part in the cell. It can divide by itself from taking commands from itself. So it does have a certain signal. It does receive a certain signal when there is time for reproduction. But the thing is, when the cell is dividing, it does not necessarily mean that mitochondria is going to be dividing too. Yeah, it divides on its own and it divides independently. Another thing that I told you just is embryosymbiotic theory, which is it's called it's also a endosymbiont. And sometimes what happens is our cell, which is this eukaryotic cell, which is very happy after the wait, is my screen, why is my screen? Okay, so yeah, sometimes what happens is this certain eukaryotic cell, inside it living are other endosymbionts, and this used to be very happy, and now what happens is it is hungry for a long time, and now his mood is changing for a bit. After 10 days it was like that, and after 15 days it's like that, and after 10 days it's gonna be like that, or kind of like a Dracula. So in the times of starvation, when eukaryotic cells are not finding enough resources, enough ATPs, or enough food, what it does, it sends what it used to do in the past. It sends certain specific, you can say, structures that we have known as lysosomes towards this mitochondria, or you can say towards this mitochondria, and they attack it and digest it, and the resources are used in the metabolism of cells for the survival of cell. So sometimes in starvation period of cell, it digests its own mitochondria by the help of lysosomes to provide energy to other parts of cell, or you can say important areas, or for the survival of cell it's important. So this was mitochondria. There are certain concepts which are apoptosis and cell digestion, which we'll be discussing in maybe tomorrow's class. Maybe, hold on, how many? Yeah, so the class has been for 50 minutes from now. What am I saying? Anyways, so today what we have done is we have understood the basics, the key difference between them, the structure of cell, and mitochondria. Tomorrow we will cover cytoplasm, ATS ribosomes, cell wall, endoplasmic reticulum. These four we're going to be discussing in the next class, which is going to be tomorrow and day after tomorrow. No, sorry, we should just do chloroplast after mitochondria. It's going to be more easy. So tomorrow we're going to be doing chloroplast. Then we're going to be doing we have done mitochondria. We're going to be doing cytoplasm. We're going to be doing endoplasmic reticulum, and after that we're going to be doing Golgi apparatus. And the day after that, which is going to be most probably Saturday, we're going to be doing ribosomes, cell wall, cell surface membrane, and nucleus is going to be at the last. And in the fourth class, which is going to be the last, which we're going to be revising all that. We're going to be covering centrioles. We're going to be talking about the basic concepts, and we're going to be talking about the last overview of the peroxisomes, glyoxysomes, and other kinds of organelles. So anyway, stay tuned till that, and we'll talk about it, and I'll meet you tomorrow. So bye bye.